Whoa, it was good. Was that? It's time for our next past paper? Yes, it is. Unit 1, Paper 2, June 2017. Did we get started? Question 1. Figure 1 is a stained animal cell as seen under a light microscope. So you're seeing this thing here, and you see magnification is times 1,000. In the box below, make a detailed drawing of the cell in figure 1 and label 4 structures seen in the cell. Alright, so what I did here is put the drawing. So you can take a look at that, and I also correlate at least 4 structures that I can see here. So we can see here, well, you know, the general jelly-like substance around it is the cytoplasm. Of course, this larger structure that you see in here is the nucleus. We have the nuclear membrane around it, and of course we have the cell membrane or the plasma membrane surrounding the cell itself. You can't really tell anything else from this magnification and from this light microscope. Everything else looks a little too grainy, so you don't want to label things like mitochondria. Part 2, using the magnification provided, calculate the actual size of the cell, highlighted by the bar line at the left of the image. State the calculated value to the nearest micrometer. Show your working. So if you measure the length of the image going up, you'll actually get 60 millimeters, which is 60,000 micrometers. The length of the cell, right, will be the image divided by the magnification, and the magnification, if you remember, was 1,000. So 60,000 micrometers divided by 1,000 is 60 micrometers, and that is your answer there. Here we have a question that's a little longer, but it's not difficult at all. An experiment is conducted to determine the amount of reducing sugar and starch in bananas at different degrees of ripeness. Extracts of equal quantity of tissue samples of bananas at 3 different stages of ripeness are prepared by mashing and grinding the samples in water. For each stage, sample extracts are placed in 2 sets of test tubes, and a 4th test tube was set up with only water and no banana extract. Of course, there's the control. Tests for reducing sugar and starch are conducted, and the findings you can see here. So let's look at the findings below. Here we have the food tests, and we have filling solution, and this tests for the presence and intensity of reducing sugar. Iodine test for the presence of starch. And filling solution, we could see here it has all these 2 food tests has been tested on 4 samples: green raw banana, half ripe banana, and water with no banana extract. Of course, this is the control. So filling solution, which is naturally blue, this would remain blue because there's nothing for it to react with in the control, and there's no precipitate being formed. The precipitate actually gives the idea of how much reducing sugar there is, and water there's none. Iodine is normally a yellow, orange, brown color, so this would have no change whatsoever with the control. However, what we could notice here with the iodine is that it has changed from this brown color to a blue-black color in all of them. The most intense being with the green raw banana. What does this mean? This means that the highest concentration of starch is in the green raw banana, and the lowest concentration of starch is in the ripe banana. The height of the precipitate is going to tell you the degree of how much reducing sugar there is, and we could see that aside from the control, the lowest amount of reducing sugar is going to be in the green raw banana, and the highest amount is going to be in the ripe banana. Therefore, a conclusion we can make is that all of this starch that would have been in the green raw banana as ripening occurs, all of that starch, and remember, starch is a polymer, which means that it's made up of a string of monomers linked together. That starch would have been hydrolyzed, or would have been broken down into reducing sugars. So this is why the intensity of the color would have decreased with iodine, because there's less and less starch, and there's more and more reducing sugar as ripening occurs. So let's look at the experiment here. So just a hypothesis for this experiment. Green bananas contain more starch and less reducing sugars compared to half and half ripe bananas. So this is what, remember, hypothesis would have been a statement that you would have to prove true or false. This would have been before you even do the experiment, and this would have been something that you would approve. It doesn't have to be a true statement. Our next hypothesis would be starch is converted to reducing sugars during ripening of bananas. In this case here, both these hypotheses would be accurate looking at the results of the experiment. The next question here is compare the results obtained for the amount of reducing sugar and starch in the banana samples. Include in your comparison reference to the observations recorded. So I just spoke about it, and I'll put up the table so you can see what I'm talking about. But it says shown by the presence of the red precipitate, reducing sugar is present at all stages. This is true because as you can see, there is a precipitate, aside from the control, there is the presence of a precipitate at all stages, just not the same amount. So that means that there is reducing sugar in all of them. Shown by the presence of a blue-black color after exposure to iodine, starch is present at all stages as well. True as well, because you have noted a color change from brown to blue-black, though different intensities of blue-black. But it still means that different amounts of starch is present at all stages of ripening. As ripening occurs, the amount of starch decreases, which means that the blue-black color intensity decreases, and the amount of reducing sugar increases, and this is shown by the height of the precipitate increasing. Once you have about, 2 of these points well explained, you should be able to get your 3 marks. Part 3. Comment on the significance of the findings of this experiment in relation to the use of bananas as a food item. So what this shows is that bananas you can eat them 2 different ways. You can eat them green, or you can eat them ripe. And riper bananas would contain more reducing sugar, and they would be sweeter and they would be softer. A lot of the the cell walls would have been broken down. There would have been a substance called pectin in the cell walls, and that's why the banana actually becomes softer as it becomes riper. That's why you can bite into it, and thus it becomes easier to consume and digest. Green bananas, on the other hand, would be more suitable for cooking, and they wouldn't be as sweet, but they would be tougher, and they would contain more starch, less reducing sugars, a little more fiber, because they would still have a lot of the cell walls, the integrity will still be there. They would be good for diabetics to consume after cooking, of course. State the purpose of testing a sample with no banana extract. Went through already, that is a control. Question 2. Hemophilia is a recessive sex-linked condition in humans in which the blood does not clot properly. This leads to excessive bleeding. Use the symbols X capital H for the normal allele and X common H for the hemophilia allele in your responses. Just to briefly go through this here, if you have a capital letter, that means that allele is dominant to whichever one is the common letter. So that means that the normal allele is dominant to the hemophilia allele. You'll also notice that the letters are not by themselves, they are in superscript to X. The X in this case is an X chromosome. X chromosomes are found in both males and females, but for a male would also have a Y chromosome, and a female would have 2. Now, the Y chromosome does not carry this X allele. It does not have the locus or the position or the space to carry the hemophilia or the normal allele for not having hemophilia. So only the X chromosome has it, and this is why it is called a sex-linked condition or sometimes an X-linked condition. So state the genotypes of the following. So a normal clotting male, basically a male who doesn't have hemophilia, will be X capital H Y. And the Y, you could put a little dash here, or you could leave it just as it is, but you're not going to put H here because the Y chromosome cannot carry the normal allele or the hemophilia allele for the disease. A normal clotting carrier female. So this female would be XX, but since she's a carrier, which means that she is heterozygous for the disease, which means that she does not suffer from the disease, but she can pass it on to her offspring. So she will be X with a capital H, X common H, with the normal allele being dominant to the hemophilia allele. Using the genotypes stated in 1 as parental genotypes, construct a Punnett square diagram to show how hemophilia-affected offspring can result from normal clotting parents. State the phenotype of all offspring. Just so you remember, what a Punnett square represents is not that these 2 people are going to have 4 children, but this would be 4 possibilities, right? Or the ratio out of 4 of what the children would be. So we're going to put the father here, and the father is going to be a healthy male, so he's going to be X capital H, and remember he has a Y chromosome that does not carry any hemophilia allele, and the mother is a carrier, so she's going to be X common H, X capital H. So we're gonna do these as 4 possibilities. So this first possibility, you're gonna inherit both X's here and both H alleles that you're seeing here. So this child is gonna be X capital H, X capital H, and this child is a girl, is a female, and she does not have hemophilia and she's not even a carrier. This second possibility here is going to be a male child because you're going to get X and Y. It's going to be a male child and he's going to inherit this X capital H here. And this child, this boy, is not going to have hemophilia. He's not going to be a carrier either because remember he only could have one allele. Y doesn't carry one. This third possibility here is going to be a girl because she's going to inherit this X and this X here, and she's going to inherit X capital H and X common H. This girl is not going to suffer from hemophilia, but she's going to be a carrier just like her mother. This last possibility here is going to inherit this X chromosome and this Y. And of course the Y doesn't carry any alleles for hemophilia, but he's going to inherit this X common H from his mother. And this child here is going to be a hemophiliac. He's going to suffer from hemophilia. Give an explanation as to why a man with hemophilia cannot pass on the condition to his son. So when we were saying this child would receive an X chromosome or a Y chromosome, those are from the gametes. And sperm cells can either have an X chromosome or a Y chromosome. And egg cells, which are the female gametes, can only have X chromosomes. So a boy would have to inherit an X chromosome from his mother and a Y chromosome from his father because females do not produce Y chromosomes. So the Y chromosome can only come from the father. And the Y chromosome, if you remember, does not contain the locus, or it cannot actually fit the hemophilia allele onto it. The position for it is not there. So the allele for hemophilia is sex-linked and is found on the X chromosome. Sons of men would receive the Y chromosome from their fathers and thus not the X chromosome with the faulty hemophiliac allele. A geneticist crossed 2 types of pea and obtained 5,474 plants with round seeds and 1,850 plants with wrinkled seeds in the F2 generation. You are required to use the chi-square test to show that these results are consistent with the 3:1 ratio normally expected from a monohybrid cross. So the first question here is state the null hypothesis for this test. A null hypothesis is basically you are assuming that there is no difference between what you are going to observe and what you're going to expect. Now, why I usually carry out a chi-square test is to see if there is any external factor that is affecting some kind of result. So, for example, if I roll some dice, if I roll a die, I would expect the chances of the die to land on 6 to be 1 in 6 because there's only 6 sides to a die. Now if I roll that die and I keep getting 4s, much more that I'm getting 1s, 2s, 3s, 5s and 6s, it means, well, something is probably wrong with the die. It's probably a loaded die, you know. And so in this case here, you are trying to see that there's nothing else other than pure chance that is affecting the results. So the null hypothesis is going to be there's no significant difference between the observed and the expected numbers. So what we expect, we should observe, and there should be really no significant difference. Now, if there is a difference, between what you observe and expect, that means that some other factor is coming into play. There may be something in the environment, or it may be some mutations occurring, or it may be some other allele that is influencing this allele, something multifactorial, and that's where we would have to further examine the situation. And that there would be what we call the alternative hypothesis, where there is a significant difference. But first we assume that there is not, and that is the null hypothesis. And what we would have to do is either reject this null hypothesis or fail to reject it, which is basically like to accept it. So part 2, calculate chi-square using the formula chi-square, which is the sum of the observed minus the expected square divided by E, where O is observed, E is expected. Show your working in tabular form as follows. So don't get frightened when you see all of this. It is not that bad. First of all, we are already given the observed and expected values, and that would have been right here, 5,474 and 1,850. How do we know the expected values now? So the expected values we can determine from this ratio. So when you calculate it from the ratio, and I'll show the calculation, we would get 5,493 and 1,831. What you would have to do is, of course, find the sum of both of these and then work it out as 3:1. When you calculate the difference between the observed minus the expected for the top, you will get negative 19. So 5,474 minus 5,493 minus 19. And if you do the same down here, observed minus expected, you get 19. When you square this value, you're going to get 361 in both cases here. And then when you take this 361 and divide it with the expected number, so in this case here, this would be 361 divided by 5,493, we are going to get 0.066. If we do the same thing here, 361 divided by 1,831, we are going to get 0.197. And our chi-square value is going to be the sum of these two numbers here, and I rounded it to 0.26. So the next part here is use the data in table 2 on page 10 to make an inference based on your calculated chi-square value. So our chi-square value is 0.26. So let's look at this graph here and try to make sense of what's happening. Now you're going to see that the first thing that we have here is degrees of freedom, and this is 1, 2, 3, 4. Degrees of freedom is basically the number of phenotypes that we're dealing with minus 1. In this case here, we only had 2 phenotypes, which was round seeds and wrinkle seeds. So our degrees of freedom is only going to be 1. And our chi-squared value, if you remember, is 0.26. And so we actually not looking at any of these other rows here. We are only looking at this first one because we only have 1 degree of freedom. What we want to look for here is something called the p-value or the critical value. So the critical value in this case is 5%, right? 5% percent deviation, or 0.05. And we could see that the value for this is 3.84. What we want to make sure is that our chi-square value is less than that. And our chi-square value is much less than that, right? It was 0.26. So that means that we're in the safe zone. So all of these here, this is safe. And anything here is unsafe. So our chi-square value is much 0.26. It's much smaller than the critical p-value, which is more than 5% or 0.05, and that turned out to be 3.84. Therefore, what we're going to do is say that the null hypothesis will be accepted. That means that there's no significant difference. Let's look at C. The variation of an organism in a population is depicted in figure 2. And we can see at extreme ends, this would be like very small organisms of these very small members and very large members of the species. And we can see that the average size would be here, and most of the individuals would be of average size. And it's kind of like that with humans too. You have some very tall, this would be like tall people, for example. This would be shorter people, and this would be average height. Now this is using the axes provided in figure 3 illustrate the effect of disruptive selection on the population distribution in figure 2. You'll see it looks kind of like a M, and to explain what this means is that in disruptive selection, it is when the organisms that are on the extremes, those are the ones that are favored. So you'll see that the number of individuals of smaller members of the species and larger members of the species are actually more dominant than those that have the average size of the species. So that might be kind of weird, but I'll give you an example here. If we use salmon or Chinook salmon as an example, now remember, for organisms to survive, they have to reproduce. And fish tend to reproduce by fertilizing the eggs externally. So basically they have to kind of deposit their sperm in, think of it like a nest of eggs. The small ones can easily and stealthily just kind of sneak through and fertilize the eggs without getting detected. The big ones, you could think of it as they can fight their way through because they're big. The average ones now, they are a little too big, which means that they get detected. They're bigger than the smaller ones, which means that they get detected easily, but they're not as big as the big and strong ones, which means that they can't really fight. So the average ones are just average. The small ones can sneak in and fertilize the eggs. The big ones can kind of rough up the way towards the eggs and fertilize them. The average ones don't have either of those advantages. This is why the population number of the average is smaller here, and this is called disruptive selection. Part 2. Using Darwin's theory of natural selection, explain how disruptive selection can lead to the formation of a new species. I just explained it here. So using salmon as an example, there can be small, intermediate, and large sizes. This is due to natural variation in the population. It's just due to having a large gene pool, a large amount of alleles that can be spread to offspring. Intermediate sizes are selected against due to the conditions favoring the other two extremes. So the small can stealthily fertilize the eggs, the large can physically fight better and thus fertilize the eggs. The average ones can't. So the successful small and large individuals would reproduce and pass on their traits to their offspring. So the small man and the tall man get through, but the average height man here get through. So he wouldn't be having much children. The intermediate individuals do not. New species may arise if the genetic variants can no longer interbreed. This is called speciation. So for example, if we end up with a bunch of small salmon and a bunch of large ones of the same species, eventually they're gonna genetically diverge so much from each other that they won't even recognize each other as mates anymore. They might even be able to mechanically reproduce anymore, and thus they would undergo speciation and become two different species. It's the same as the finches on the Galapagos Islands. Let's look at question 3. An experiment is conducted to investigate the rates of type of culture medium and sucrose concentration on pollen germination rate of a species of palm. Pollen grains are collected from newly opened flowers of the same bunch on the same day. The grains are inoculated into two types of medium. So you have a liquid culture and a solid culture. The solid culture consists of different concentrations of sucrose in agar, that's a type of gel. The liquid medium is similar to the solid in that it has sucrose in it, but it does not contain the agar gel. The results of the investigation are given in table 3. In the table here, we can see the sucrose concentration in grams per liter, and we can see that, well, this is going up, is increasing, and we are testing for the percentage pollen germination as the sucrose concentration increasing. Just from a quick glance on the table here, we can see from the liquid culture that the peak germination rate is, which is 73%, is at 40 sucrose concentration, and basically when it reaches 100 sucrose concentration, there's no germination at all. There's zero, none. So something wrong here with the liquid culture. Solid culture, on the other hand, we could see that the peak is again at 40 grams per liter, and that's 87% germination. And you could think of it as it keeps going up. It peaks here, and then it sort of goes down, goes up, goes back down. So it sort of fluctuates after 40 grams per liter. Let's look at the graph. Here you'll see the graph depicted, and your marks here are simply to label your axes correctly. Make sure that the percentage pollen germination, which is a responding variable, that there will be on the y-axis, and your manipulated variable sucrose concentration is going to be an x-axis. Make sure you put a unit, grams per liter. This will be a line graph, and since there are two media being observed, liquid and solid, you want to make sure that you draw one with a solid line and the next one with a dotted line, or that you use two different types of points when you are plotting. So you could do one with a X, and you could do one with a circle, with a dot with a circle around it, sorry. Or you can make sure to just label the ends of each one. In either case, you need to have some kind of key or legend to indicate which one is which. Now from the graph, what we could see is that the liquid medium, as sucrose concentration increases, this is the solid line we're looking at, this is going to increase, is going to peak here at 40, and then it's going to steadily decrease until it reaches 0% germination at 100. The solid medium here, we'll see that at lower sucrose concentrations that the pollination rate is lower, but after 20 grams per liter it has a sharp increase, a steep increase, until it peaks at 40 grams per liter, and this is about, we say, 88%, and then after that it begins to fluctuate. Briefly describe the overall trend for the effect of different sucrose concentrations in both culture media on the germination rate of the palm pollen. Is basically what we just described. You want to put at least 2 points for both liquid and solid culture media. Germination rate increased with increase in the sucrose concentration until it reached a maximum rate at 40 grams per liter. After that, it basically started to fluctuate or decrease. Beyond 40 grams per liter, the germination rate decreased as sucrose concentration was increased. Easy, there's a trend there. Based on the results shown in table 3, what can be deduced about the effect of the type of culture media on the maximum rate of pollen germination? We could deduce that the solid culture media works faster for germination compared to liquid, because you'll see that there was the steep increase there. The liquid culture medium has higher germination rates at sucrose concentrations beyond 40 grams per liter than the solid culture medium. B. Figure 5 is a drawing of a section through a human placenta in situ. And that just means while it is still in the uterus, it means on site. And we could see a number of things being labeled here. And they said identify and describe the main function of each of the structures labeled 1, 2, and 3. 1 is going to be the chorion or the chorionic villi. And what these do is they facilitate exchange of materials between the maternal and the fetal blood supply, and it also acts as you can think of it as it also acts as a buffer to the mother's blood pressure, where the mother's blood pressure would be way too high for the fetus, so it actually decreases the pressure as it comes through to the fetus. What would happen here is that certain materials would be able to diffuse through these villi, for example glucose and oxygen, whether it's through simple or facilitated diffusion, and substances would also be able to diffuse out, for example carbon dioxide and urea. 2 would be this could work as uterus lining, uterine lining, or uterus itself, right? Remember, the uterus lining is also called the endometrium, and what that does is that it holds the fetus in place. If you talk about the uterus lining, it supports attachment and development of the placenta and fetus when the blastocyst attaches to the uterus lining, it sort of digests a little pocket in there so that it can actually connect both blood supplies, so the fetus can get rid of waste and it can receive materials from the mother. The last thing here is 3, which is the umbilical cord, and the umbilical cord is basically the lifeline of the fetus to the mother, and this connects the placenta to allow delivery, for example glucose or removal materials. So this is exactly what facilitates the exchange, and the villi is what brings it towards the umbilical cord in the first place, the materials. Part 2. Comment on 2 roles of the structured label Y in the development of the fetus. So Y was the amniotic sac or the amnion. And what the amniotic sac does is, imagine it's like a shield for the fetus in case the mother gets a little hit on her belly. The fetus would not, it would not receive that same force. So it acts as a shock absorber to protect against mechanical damage. But it's not just that, it also acts to keep the temperature of the fetus stable, because most of it is made out of water, and water has a high specific heat capacity. So it basically absorbs a lot of heat, prevents the baby from overheating. It also acts as a reservoir to nutrients, and it helps to support the developing fetus against gravity and holding it in a safe position. What it would also do is that it would support the movement of limbs. The baby is basically swimming and limbs are developing, and it also engages the swallowing reflex in the fetus. Next one, outline 2 ways in which the placenta acts as a protective barrier for the developing fetus. What the placenta would do is that it prevents large molecules from entering the fetus's bloodstream, such as maternal hormones, basically anything that would be incompatible with the fetus, or might not work well for development of the fetus, or might be detrimental to the fetus. So it would limit large steroid hormones and so on from actually entering and going into the fetus itself. It would also reduce the blood pressure of the maternal blood. I just spoke about that, as materials enter the fetus. And the placenta also delivers antibodies to the fetus to facilitate natural passive immunity before, at least for the first years of its life, so it can be able to protect itself against pathogens, at least until the baby is old enough to get shots. Question 4. Compare the cell size and 3 structural features of prokaryotic and eukaryotic cells. I have 4 points here. 1 relates to cell size, where eukaryotic cells are much larger than prokaryotic cells, as we have to note there. But eukaryotic cells, which are typically found in protists or multicellular organisms such as plants, fungi, or animals, those are of course more complex than prokaryotic. Prokaryotic is where you'd find in bacteria. And the 3 differences that you'll see here is 1. Eukaryotic cells contain double membrane organelles such as mitochondria and chloroplasts. Prokaryotic cells don't have these double membrane organelles. 2. Eukaryotic cells contain a nucleus, or what you call a true nucleus, while it is absent in prokaryotic cells. What prokaryotic cells would have instead, of like bacteria, they wouldn't have a nucleus, but they would have a nucleoid region where nucleus material, DNA material would be stored in circular form or in strands, kind of like chromatin. 3. The size of the ribosomes. Eukaryotic ribosomes are larger, and they are called ATS ribosomes, than prokaryotic, which is 70S ribosomes. The S in this case stands for sievert, and think of that as a type of unit that relates to density and how dense materials move through a medium. Part 2. Outline the endosymbiont theory for the origin of eukaryotic cells. Explain how characteristics of mitochondria and chloroplasts provide evidence in support of this theory. So the endosymbiont theory, if you think about the word endo means inside or in. Symbiont comes from symbiotic. You think of it like symbiotic, which means that it has a sort of relationship, and in this case kind of like a mutual relationship. So what it says is that mitochondria and chloroplasts were once prokaryotic organisms, very early prokaryotic organisms. What happened is that those would have been, when the earth was very, very young, those would have been like swimming in the ocean, and what they would have done is that they would have invaded a larger host cell. And the early mitochondria, they was able to undergo respiration. The early chloroplast was able to undergo photosynthesis. So this larger host cell now would get those two, think of it as like it gaining the powers of these two. It now is able to respire and photosynthesize, right? And this larger host cell would basically be like a blob of cytoplasm that wasn't able to do much by itself. But the mitochondria and the chloroplast would have integrated within them symbiotically, and thus would have formed what we know now as the first eukaryotic cells, which would have then evolved into much larger complex organisms like horses, cats, dogs, and humans, and plants, and fungi, and so on. So what is the evidence for this? We want to provide at least 3 or 4 points of evidence, and the evidence is this: both organelles, mitochondria and chloroplast, are actually similar in size to prokaryotic cells. A lot of them are the same size as some bacterial cells that we know today. Both organelles also have the same circular DNA, similar to prokaryotes, but it wouldn't make sense for the mitochondria and the mitochondria and the chloroplast to have a nucleus, but they do have DNA. They have their own contained DNA. Both organelles were produced by binary fission, similar to prokaryotes, so they divide, just like how a bacteria parent cell would divide into 2 cloned daughter cells. Lastly, ribosomes within mitochondria and chloroplasts are of a similar size, that small 70S size, similar to prokaryotic ribosomes. So even though mitochondria and chloroplasts are found in eukaryotic cells, they have the same type of ribosomes as in prokaryotic, which is interesting. Alright, let's look at B. Discuss the importance of protein structure for determining enzyme specificity and mode of action. Basically, what makes an enzyme specific to the substrate it wants to react upon, and how does it work? That's the mode of action. First, we know that enzymes are globular proteins that have tertiary or 3-dimensional structures. They are catalysts for biological reactions. Of course, common examples would be like pepsin, trypsin, amylase, lipase, and so on. They do this by lowering the activation energy for the reaction. So I'll put up a little graph here, and what this means is that you'll see the very steep part, that would be the normal amount of energy required for the reaction. Of course, this would take a longer time to achieve. What the enzyme does is that it lowers that amount of energy. So think of it as a large mountain turning into a small hill. It's easier to climb the hill than to climb the mountain. It takes a shorter amount of time. Thus, what it does is that it speeds up the reaction. Enzymes contain active sites. Active sites are regions of specific size and amino acid sequences for the binding of substrates. The active site is basically, think of it like a lock and a key, and so it has to be specific to fit, just like each key is specific to a lock. And next way you could think of it as like a hand to a glove. The thing is, sometimes when you put on the glove will kind of fall over your fingers, and it would fit in a certain way. That is called an induced fit, where the enzyme might actually change shape a little bit to accommodate the substrate. So what happens here is that the substrates bind to the active sites due to their complementary shape, similar to a lock and key mechanism. After binding, you get an enzyme substrate complex, which is the combination of both of them, and the enzyme can slightly change shape in response to binding, like a hand and a glove. And this is an induced fit. (5a) Describe the roles of DNA and RNA in protein synthesis. DNA is a nucleic acid made up of nucleotides. Nucleotides contain bases, sugars, and phosphate groups. And those bases would be like adenine, cytosine, guanine, and thymine. That's in DNA. And what those would do is that those would link together in long chains and long sequences and can be read. Those letters can be read in triplets, and we call those codons. But basically what that DNA would do is that DNA, any sequence of that would be called a gene. And a gene would be like a blueprint or instruction to form a protein. So for example, if you have darker skin, you would have more protein in your under your epidermis called melanin, and you would have your DNA would be the blueprint to produce more melanin to keep your skin dark. Same thing would be for keratin and collagen, or any protein that you could think about, including enzymes. So there are several types of RNA involved in protein synthesis. So there's mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. We want to talk about all three. So mRNA is used for a process called transcription. What transcription does is that it basically writes the genetic code from the DNA onto a single-stranded molecule called RNA. And what it does is that the difference between DNA and RNA is that RNA could move. It is mobile. So the RNA, messenger RNA, is what it is like a messenger boy that is like, take this message from me and carry it to these ribosomes and tell them to build this thing for me. So the genetic code is copied from the DNA into codons. It's like sending a message. During this process, DNA is unzipped to expose the bases, and when is exposed, what happens is that the RNA nucleotides, which it has all the same ones, adenine, cytosine, guanine, but it doesn't have thymine, and instead it has one called U, which stands for uracil. So it allows these RNA nucleotides to pair up to form complementary mRNA. So just like how you would have two strands of DNA, the mRNA is very similar to the other strand of DNA. And because it is mobile, it can actually take the information away from the nucleus to the ribosomes in the cytoplasm. Transfer RNA, now tRNA, would be used to translate this code. So transfer RNA has each one has an amino acid bound to it. And what would happen is that the ribosome would read this message from the messenger RNA, and the tRNA, in response to that message, would deposit amino acids one at a time. With each one it deposits, is going to build a polypeptide chain, and that polypeptide chain is then going to fold and become a protein, a 3D structure of protein, for example, an enzyme. So it's used to produce amino acids from ribosomes reading the mRNA codons. A polypeptide chain is produced as various tRNA molecules deposit the respective amino acids to the ribosomes. I'll put up a diagram so you'll see how this is done. Ribosomal RNA now form ribosomes. These are the constituent, think of them as they are the building blocks of ribosomes. And what ribosomes do is that they move mRNA molecules, kind of like a ticker tape timer. It reads it like a message, and that facilitates the assembly of polypeptide chains, which again would fold into protein structures. So this is how DNA eventually becomes or codes or it instructs on how to form a protein. B. Genetic engineering and gene therapy are two closely related technologies with promising potential for improving human health. Outline the principle of genetic engineering, including a brief description of the basic steps of this technique. Genetic engineering involves the transfer of particular genes from one organism and placing it into another, thus altering its genome. It can involve the alteration of an existing gene. So for example, if someone has cystic fibrosis, the genetic engineering can help remove the faulty gene and replace it with the one that actually is supposed to be there. Common example of genetic engineering would be glowfish. So when the gene from a bioluminescent jellyfish is placed in a zebrafish, when the zebrafish's muscles contract, they glow. So that's why it could have all these pretty colors. So what's the steps to doing this? So step number 1 is to identify and isolate the gene using restriction enzymes to cut DNA. Restriction enzymes are formed by bacteria when they fight off viruses. Think of restriction enzymes as the bacteria's white blood cells, if that makes sense. And what the restriction enzymes do is that they can act as DNA scissors to cut out pieces of viral DNA. And so the viral DNA won't work anymore because it's basically snipping up the instructions. Why they call restriction enzymes is because they actually restrict the virus from doing anything to them. So after isolating the gene using restriction enzymes, you can glue them together to a vector or plasmid. A plasmid is a circular piece of DNA. And what would happen is that imagine that you would take that little piece of SNP DNA and glue it on to this plasmid using DNA ligase as an enzyme, and think of that as genetic glue. So now this is going to form recombinant DNA. Recombinant DNA is basically the plasmid now would contain the DNA, let me say, from a bacteria, and it would also contain the gene that you want to put into something. What you'd have to do now is introduce the vector to a host cell, for example, E. coli. So you could sort of mix the bacteria with the plasmids and hope that they take them up. Whether they take them up or not, that's a whole different story. But you're going to see if they could take them up. Now, the ones that do get the bacteria that do take up the plasmid, or the host cell that does take up the plasmid, you just leave them to undergo binary fission so they can clone and they could reproduce. So if you only get like 10 of them taking up the vector, at least over time, if you give it some glucose and oxygen, you might end up with millions of them as they undergo binary fission. Part 2. Despite its promising potential, one of these arguments sometimes raised against the use of gene therapy is that it is technically too dangerous. With reference to cystic fibrosis, discuss reasons for this viewpoint. So you want to discuss about 2 reasons, because we see here that this is 5 marks, so 2 reasons well explained, and it must be in relation to cystic fibrosis. So the first one here, 1. Viral vectors may result in infection and inflammation of the lungs, especially if the patients are immunocompromised. Now typically with cystic fibrosis, you would use a virus to transfer the DNA into a human. Why a virus? Because a virus can penetrate the cell membrane very, very easily. The problem is that it's a virus, and viruses are bad, and they can mutate, and they are unpredictable, and they can multiply very easily once again to a cell. So typically the virus has to be inactive. You never know if it could reactivate. So the viral vectors may also mutate in a patient, and death has been noted in actually a few cystic fibrosis treatment patients. 2. The therapy will only work if the gene is delivered to a large number of tissues. I say one of the problems is that if you will be able to deliver this gene to enough cells, sometimes the gene will be delivered to cells that don't need the gene at all. Or sometimes it becomes trapped. In this case with the lungs, because cystic fibrosis affects the lungs, there's a lot of things that prevent viruses from getting into the lungs. There's cilia and there's mucus. And when you have cystic fibrosis, you produce a lot of mucus. So the vectors may become trapped in those, and the vectors may deliver DNA to non-target cells, the ones that don't need help, and thus may be inefficient. This should be okay to get your marks here. Let's look at question 6. A. Given examples, discuss 2 advantages of vegetative propagation of plants. So vegetative propagation is a form of asexual reproduction, which means that you only require one parent. And what happens here is that there's no production of gametes, so it allows new plants to be produced from processes such as grafting, budding, and cuttings. So you could cut a piece of a stem of a plant, put some rooting hormone, put it in soil, and that stem will actually grow adventitious roots and become a new plant. A good example would be sugarcane. So it allows rapid multiplications of plants of identical genotype, because there's no fertilization or anything involved. So it can, especially with things like tissue culture, if you're using cuttings, you can do a lot of them at a time. And there's no mixing of alleles, so you maintain favorable characteristics and ensure that they are well adapted to stable environmental conditions. Tissue cultures ensure plants are also disease-free because they are grown in sterile conditions. For some species, such as bananas, it is the only viable method of reproduction. This is because bananas, the seeds are actually not viable. They can't plant a banana seed, well, at least for a domestic banana, and expect it to grow. Bananas are reproduced via cuttings. You could also use grafting to mix the alleles of two fruits, for example, or two crops. So you have some oranges, you have some mangoes that the variants are actually produced by grafting one stem on the other. I'll put up a little diagram to show you what grafting is. The grafting can also be used to produce hybrid versions of plants with mixed characteristics. You can't really rely on mixing of alleles via gametes because there's no gametes involved in asexual reproduction. So B, discuss 2 genetic consequences of sexual reproduction. So sexual reproduction leads to increased variation in organisms, ensuring high biodiversity in populations once you have a large enough gene pool and you have a large population. This allows members of the species to overcome selective pressures such as pests and disease, and possibly facilitating speciation. Again, Charles Darwin and the finches on the Galapagos Islands are very good examples of this. 2. The formation of seeds during sexual reproduction improves distribution of members away from parent plants. So we know that there's seeds that can be transported very far from the parent plant. For example, seeds that have parachutes and are transported by wind, or seeds that are buoyant and can be transported by water, or seeds that can be eaten by birds and transported very far away from their parent plants, when the seed could be broken down in the digestive system of the animals or the birds. You have seeds with hooks that can latch onto animals' fur. So what this does is that it reduces competition, and it also increases the chances of colonizing new areas where you wouldn't usually find those plants. Examples would be how coconuts ended up on beaches. They floated there and got planted there, and they grew from there. And this is the last question of this paper, part C. Describe the development of fruit and seed structures from floral structures after fertilization. You want to list 6 things here because it's 6 marks. So this is post-fertilization structures. So this would have been after the whole pollen tube thing would have grown, and after the 2 male nuclei fuse. So we're talking about after that. So remember, the egg would become a zygote. So the zygote is formed, and eventually that zygote is gonna undergo mitosis and become an embryo. Next we have the endosperm, which you remember is formed from a triploid endosperm nucleus. And the endosperm is, think of it as the plant placenta. It is basically the food storage, and that will supply nutrients to the embryo. Now, when the embryo develops, we're going to have a large basal cell and a suspensor cell column that attaches the embryo to the ovule wall. It sort of looks like an umbilical cord. The embryo, as it grows, is going to develop an early root called a radicle, an early shoot called a plumule, and very early leaves, which will help it photosynthesize in the first days of its life, called cotyledons. The ovule is going to develop into a seed, and the ovary develops into a fruit. So let's say that we have an apple flower that gets fertilized. Eventually that is going to become an apple fruit. The ovary wall develops into the pericarp, or the flesh of the fruit. So for example, if you bite into a mango, that sweet thing that you taste in is the pericarp. If you ever eat coconut jelly, guess what? Eating pericarp. And if you drink coconut water, nice little fun fact here, you're actually drinking coconut endosperm. That was actually supplying nutrients to the coconut embryo. You learn something new every day. Anyway, that's it all here. Take care. Laters.